first of all, thank you all for coming. The partnership that we've had with Alliance for Regenerative Medicine has been really important in trying to have us move products forward, to partner with industry to see these advances actually make their way from concepts through production to patients. So what I want to do today is tell you a little bit about some of the CAR T therapeutics. There's been obviously some modest developments that people are interested there. Talk a little bit about rare disease therapeutics, what we're doing in that area to try to help move forward a place where we have stagnated a bit. And with that, tell you about the state of the field of gene therapy, and then talk a little bit about how we plan to try to help address challenges. I'll say from the outset, we don't have the answers for all of the problems. We can't. On the other hand, we are going to try to do whatever we can here in the interest of patients. So right now, we are sitting in a nice place, 17 approved gene therapies. You can see the code here. Some of these are CAR T-cell therapies. Some of these are genetically modified stem cell therapies. And 7 of them are directly administered gene therapies, starting with Luxturna the first, which was locally administered, and others that are either locally or systemically administered. So really, this list has built nicely. So what I'm going to do now is just focus first on some of the CAR T-cell issues. So CAR T-cells have been remarkable in the area of oncology. I'm a hematologist-oncologist by training, and to be able to salvage children who had refractory acute lymphoid leukemia and have children now that are long-term survivors because of this therapy is really remarkable. And they're surviving with actually remarkably low amounts of toxicity from the therapy long-term. It doesn't salvage everyone, which means we can always do better, but it's really quite remarkable. You see the survival curve there for acute lymphoid leukemia in children and young adults. And actually in adult non-Hodgkin's lymphoma, also a reasonably good salvage rate. Multiple myeloma, the survival curves might not look quite the same, but it's a harder nut to crack potentially. So in all of these, though, now, hematologic malignancies, and a tremendous amount of progress because of these therapies. If you look, though, there are some safety concerns that can occur with these products. And we've known all along that we've had to deal with cytokine release syndrome, and managing that was part of getting the first therapies approved. And the initial companies that were putting these forward and moving these forward really had to do some trailblazing work dealing with kind of pathways to manage the administration of these therapies and manage the toxicities. We have this issue of some neurologic syndromes that can actually develop in some of these. And then there are some various other side effects. All in all, though, they're generally manageable. And we have known all along that because these are administered through either lentiviral or retroviral vectors, we do have to follow people because there is obviously the concern that there could be some type of genotoxicity. And what did happen in the months leading up to November were some reports of T-cell malignancies that came into FDA, variety of different types of T-cell malignancies. And as of the end of last year, we had 22 cases reported. Now, we don't have full data, as we often don't, as things evolve on all of the cases, and we're still working to get more data. But although sequencing is not available for all of them, in at least a few of the cases, we know that the CAR construct is in the malignant clone, which really suggests that there was probably an association there. And the onset of these was soon enough after the administration in some cases that it does look like there's a causal relationship. That said, there were 27,000 plus individuals treated in the United States with these. And so the rate of a few of these cases with tens of thousands of individuals treated, it's not overly concerning to us in this malignant setting right now. Yes, we did put out a safety alert. Yes, it's going to require follow-up. But again, I think one has to step back and remember, as a hematologist oncologist, the other way to try to salvage people is to give them combination chemotherapy with cytotoxic agents that have secondary malignancy rates measured in the low percent range. So this is a risk-benefit balance. Moving forward, I think we've put out some initial guidance here about what we're asking for: long-term monitoring, asking that providers, if they see a new T-cell malignancy, contact manufacturers, contact us so that we can get the data we need to understand the frequency of these events. But just make no mistake, the overall risk-benefit profile here is still incredibly beneficial, and so for all of the approved uses, we don't have any concerns with continued use of these products. There may be some discussion that we can have at the end about uses in other and development in other areas, which I think also will be able to proceed. But this is something that, like many things at FDA, don't take what I say today to be the final word, because over the coming months, as we learn more, we will adapt and update our information. So I want to move on to gene therapy for rare diseases. So this is something that is near and dear to my heart. The importance of these diseases is, right now everyone wants to focus again. We've kind of gone it's the sine curve just keeps going. Large disease populations, small disease populations, large disease populations. These are really important precisely because in aggregate there are millions of individuals infected. Additionally, if you can't, this is just my personal opinion, if you can't get it right for small batch gene therapy, there's no way we're going to get it right for large gene therapies. And frankly, I would rather see small populations exposed first and then understand how that might have implications for larger populations. So let's think about it for a moment. Small molecule protein therapeutics, antisense oligonucleotides, perfectly good way of going about addressing rare diseases. But actually, they're potentially more convoluted than gene therapy. And why? Because generally, for gene therapy, you have this high possibility of success up front because you know what you're working with. You know the gene of interest. You know probably where it's important to have it expressed or to have less of it, and you can manipulate that. This concept of one dose or perhaps just a few doses to be able to do something is very attractive. This kind of, if you look at some of the successes, this concept that you could have long-term benefit with one dose is very attractive. But there are issues, and that's what I'm going to start to move to focus in on. There's complexity and cost of manufacture. This has become a real challenge because gene therapies have not followed the monoclonal antibody chemistry manufacturing controls model. And it does make now, for a while I was like wondering, why? Well, of course it makes sense because unlike monoclonal antibodies, which are made continuously because there's a continuous demand for them, even in people who might only get them for a year, there's another batch of people that get them for another year, some people are on them for lifetimes. Here, you've got a one-and-done therapy, so the manufacturing, you have to come up with new models there. There's the possibility of irreversible side effects. That's genotoxicity, something we have to be aware of. If one's giving these, you can't just go to the local health stop and stop in for a little gene therapy. So you have to have expert centers to do this in order to do it safely. And this really is a new business model, and the struggle about how you pay for this, how we as a society pay for this, it's not in my department, but it is one that obviously, well, it's in our department, the Health and Human Services, but it's not in my department, is a challenge. What does this look like? For those of you, many of you are probably familiar with this, but spinal muscular atrophy is a terrible rare genetic disease. Type 1 spinal muscular atrophy, kids just can't do all the normal things that kids do starting from the beginning of life. They can't raise their heads, can't sit up, can't walk, and eventually stop having the ability to breathe and they die. Although there have been various therapeutic approaches thought of, gene therapy is obviously a great idea here. Zolgensma or anasemnogene was developed, and it was very rapidly apparent when this was given early in life, this had a game-changing effect. And the child on the right here is running around at age 3. None of you need to be statisticians or anything to know that if I tell you that normally all children are dead by age 3 and you see a child running around, probably something worked. And indeed, in 14 out of 15 children in the original trial, they had an outcome like this, really quite remarkable. This is gene therapy when it really works nicely. It doesn't even have to work this nicely, though, because if you're the mother or father of a child with Duchenne's muscular dystrophy who's 15 years old and now is almost not able to walk or not able to walk, and you want to hope that they can continue to use their arms, just having something that will stabilize their disease could be a huge win. That said, we've got some real challenges here. And I actually, despite the fact that I generally am in the regulatory and the science world, business in this case can't help spill over. There has been obviously a downturn in the economy and a downturn in this area. And I think we're at a critical juncture, and I think besides, I'm going to keep aside interest rates, aside from those things, aside from investor interest, there are manufacturing challenges, issues with clinical development timelines that impact patients in part because they impact the investment community's willingness to put capital into this area, and that's something that we have to think about how we can help at FDA. And different global regulatory requirements, which create a real problem. If we have a population of 50 patients in the United States, the EMA, European Union has 75 patients, Japan has a number of patients. The 50 patients in the United States don't tip the net present value calculation into the positive. If you aggregate everyone, you have a much higher chance of tipping that calculation into a place where it actually makes sense. And so this unfortunate divergence of global regulatory requirements, which in this field of rare diseases where it's affecting so few individuals, it's actually something we have to do something about because we owe it to the patients globally. So I'll tell you a little bit about each of these. So at Center for Biologics, as I say, we may not be able to make headway on all of these, but we're going to do our best. We're going to try to advance manufacturing technologies for cell and gene therapy. We're sponsoring some research. We're encouraging collaborations with the Foundation of National Institutes of Health, the Bespoke Gene Therapy Consortium. I'll tell you more about application of the platform technology provision, which was passed in 2023. Talk about a little bit about the efforts to use accelerated approval for gene therapy and why we believe it actually makes eminent sense and why it shouldn't be controversial. And explore this idea of concurrent regulatory submission with other regulatory authorities. And then finally, talk about something good that may have come from the pandemic. So if you think about manufacturing for gene therapies, let me try to get my slides right here. If you think about manufacturing for gene therapies, we are unfortunately at a place where the best net present value calculation comes in the medium-sized cup. Okay, unfortunately, it's a little bit like Starbucks. They only put the venti up now. They don't even show you the tall or the grande. Sorry, the grande, not the venti. It's the middle size is the grande. I got to get my Starbucks lessons right so that when they kick me out of FDA, I'll be able to be a good barista. It's the grande cup that's the one in the middle. So it's that middle size that we're good at. The problem is the setup cost for these small gene therapies is just way too high in proportion to the return on investment. So we have to figure out a way to get over that. One of the ways that we can do that is try to get better harmonization. A lot of the innovation comes out of academic laboratories that use whatever technique they want to make the gene therapy. They get some data, and then they try to pass it along to industry or even to a CDMO to make more. That each time that transfer occurs, it costs a lot of cash, potentially millions of dollars. If we standardize that, that might help that. The other issue is that if we can automate at least parts of the manufacturing process, such as the downstream purification of gene therapy vectors, we might be a lot better off. And I'm sorry, it's a soda dispenser. I don't have the machine yet, but it may be that small batch gene therapy may be, the future of that may be fabrication devices with a disposables and consumables model, much like what we have now for some of the CAR T cell manufacture for the cellular products. Platform technologies are something that we are now starting to pay a lot of attention to. A lot of the gene therapies that we are thinking of have a certain difference from many of the other drug products and biologics that we use, because they have something in common, the vector backbone, that's the same between the products, and then you're swapping out inserts. And so the omnibus of 2023, we provided input, and Congress passed a provision which allows the use of this platform provision, which allows manufacturers to reference prior manufacturing, toxicology information, et cetera, from prior applications, so that hopefully as we move forward in gene therapies, it will be easier to bootstrap your way from one to the other. Now, the reason why this was actually essentially prescient is because of this. CRISPR-Cas9 is the poster child for a platform therapy. This basically shows you that since the initial CRISPR-Cas9, which was just simple editing, we now have much more complex editors and base editors and prime editors. And the thing about them that these are similar about is that what you're dealing with is a molecule that's basically the same with the exception of a guide. And so that means that even across different diseases, you're going to have something that's basically the same, where your focus, if you're a regulator, is basically on what's going to get you into problems. That is, anything that the guide can give you as an off-target effect. So these are really, we have to get platforms right if we're going to move into the era of CRISPR-Cas9 genome editing. And to be honest, just looking over the horizon, 10 years from now, if you combine CRISPR technology with lipid nanoparticle and targeting, you might get to a place where a lot of our manufacturing issues that we're having now start to go away, because you're not dealing with viral vectors the same way. But that's a forward-looking statement, and you shouldn't take that as meaning very much. Okay. We want to leverage accelerated approval, as we've said several times. And that's because when you have a gene therapy, you generally have an enzyme or a structural protein. Not always, but you generally have one that you can measure. And you can see if you've reduced it if you have too much, or you've filled it if you've had too little. And by doing so, you can do that in a variety of settings. You can do it in an animal model. You can make animal models and show that you've reduced, you have an animal model that doesn't have any of the protein. You give a gene therapy and you increase the level, and you show the animals seems to be normal again. Or you can look in humans, and sometimes there are any number of genetic diseases where you have low levels of a protein, but the person is ostensibly normal. So once again, if you can get the levels up to that, you have something that's reasonably likely to predict that the product will have clinical benefit. And that's just what we need to get forward here with accelerated approval. We want to get more things over the finish line. This is a way to do it. Will we always get it right? No. But if we get it right 90, 95% of the time, we'll be in good shape. And to that extent, we will not have kept back therapies from rare diseases where we could have helped somebody, that, where something could have made it there, and it didn't because we were just too conservative. Obviously, we have to have safety as first and foremost, but if we have to sort out some things for lack of efficacy, as long as it's not too high a rate, we can live with that. In this area of gene therapy and global collaboration, we are working. We finally have some traction. I think the pandemic probably helped this somewhat too, which is that global regulators realize that what we do is more in common than divergent. In other words, what we do at FDA has a lot in common with what's done in EMA. There are some subtle differences. We do certain things they don't do, and vice versa. But if we can harmonize on our requirements and potentially even pool forces to review these products, we may be able to make it much more attractive for people to go into this rare disease area. So this is another lever that we have to pull. And finally, as someone who spent a lot of time at the front lines of Operation Warp Speed for the accelerating vaccine development, obviously manufacturing at risk, and scaling up capacity, all of those things were really important to getting the vaccines in 11 months. But something that probably took at least 5 or 6 months off of the overall timeline was just the fact that we had constant communication with companies. You don't have to believe me. Go ask someone at Pfizer, Moderna, Novavax, AstraZeneca, Janssen, how accessible we were during that time. We had teleconference at 5 a.m., 6 a.m., 10 p.m., just to get things moved through. Now, we can probably keep things to normal business hours in this pilot, but the idea here is if we were to do this, do something similar in the rare disease space, could we help small companies in particular move things forward? I would put it to you, having seen small companies struggle, that this could make a big difference. We're going to have to show that it does, though, because no one's going to put out the dollars. It's very labor intense. No one's going to put out the dollars in government unless we show that we are making a difference. So we're doing a pilot program. We actually have time open now where we're accepting applications for 3 rare disease products in the clinic now, optimally in phase 1, phase 2, which are targeting rare pediatric genetic diseases. And we'll select them, they'll get this kind of concierge service, and we'll see how fast we make milestones, and we'll get feedback from the companies on how it works for them. So these are just a variety of ways that we are really trying to have things move so that we have a breakout year this year. I think this is a great year for us. In the federal government, I was telling my team this morning, this is a great year to focus on gene therapy, because I don't want to know what other stuff is going on, blah, 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 blah. I don't want to know it. I just want to focus on moving ahead gene therapy. Thanks so much. Questions? Yeah, go ahead.
Maybe just one quick question. You touched on the CAR T therapies and the risk-benefit profile, and other areas. If you think maybe, based on everything you know right now, looking at the non-oncology indications maybe led by autoimmune, how do you see the risk-benefit equation?
So for those who might not have been able to hear that question, the question is, for the non-oncology applications of CAR T cells, those would be autoimmunity-based applications. What do we see as the how those might go forward in development? And I first want to say that there'll probably be a little period here where it takes us to get everything straight with all the advice we give everyone. So I apologize to any company that might not have gotten this advice, but where we are settling out is that in general, the risk-benefit is still going to be favorable to move forward in those development plans. And that's in part because autoimmune diseases are not benign diseases. Anyone who's ever known somebody who's had lupus cerebritis or lupus nephritis will know that those are potentially lethal diseases. So we will, those development programs can and should continue. We may ask for some additional safety monitoring, more to come on that, but I think the important message is don't think that that should be an area that people should run from, because it's important that that continues, especially because there are second and third generations of those products potentially that could reduce the risk of random integrations using CRISPR technologies, et cetera, that may lead to continued need for us to really move forward there. And those patients with autoimmune diseases that are currently treated with pretty toxic drugs to try to address them need that potential hope for those therapies. So thanks for that question. Okay, great. Thanks very much.